way down. Now, when we spoke about force generation of muscle, what was the main concept here? What translated to more force in skeletal muscle? Cross bridge cycle, perfect. So we know that more cross bridges equals more force, okay? Now, what were the factors that we said could affect a single muscle fiber? What could we do to a single muscle fiber to make sure that it was giving us the maximum amount of force? What are the factors that we spoke about? Let's start with the action potential. How can we manipulate the action potential to give us more force? We increase the frequency, right? So increasing the frequency of action potentials also gives us more force. And here we spoke about summation, we spoke about TREP. Make sure you review those two things. What else did we say can increase force generation? What are the factors of a single muscle fiber? Where do we like the muscles, the sarcomere to be in terms of generating the maximum amount of force? At the optimal length, right? So the optimal length equals the maximum force, okay? Now, what does optimal length mean? Just to review that a little. This had to do with the way that the sarcomere was, right? If the sarcomere was too stretched out, we didn't generate the maximum force. If it was too contracted, we didn't generate the maximum force. That optimal length is that perfect in-between length that gives us the most amount of force. Now, these are the factors that can affect a single muscle fiber, right? A single muscle fiber. Oops. What did we say could also increase the force, but had to do with more fibers? What was that principle called? Not slow twitch. That has to do with the fiber types, but specifically for all muscle fibers, what can we do to generate more force? Recruitment, perfect. So recruitment, right? We can recruit or call on additional fibers. Now, we said recruitment followed a specific principle. What is that principle? The size principle, right? The size principle. And what does the size principle mean or tell us? Exactly, we recruit smaller units, right? So smaller units. See this? Before large units. Okay, and this not only had to do with the size of the unit, i.e. how many fibers were attached, but this also had to do with the size of the axon diameter, so a smaller axon, and the size of the fiber diameter, right? So a smaller fiber before a larger fiber, larger axon, okay? And that's the size principle, that's recruitment, okay? The next thing we spoke about as far as force generation was the velocity of shortening. And what were the three factors that we said were affected with the velocity of a heavier load versus a light load? We spoke about three factors with the velocity. So if I'm lifting a box that's full of books, what's going to happen in terms of how fast I can do that? So we said a heavier load, i.e. a heavy box or a full box, is going to have an increased latent period. Why does it have an increased latent period? Why would we have an increased latent period for a heavier load? What does the latent period represent? So the latent period we said was that time where excitation, contraction, coupling was happening, right? And if we need to generate more force to overcome a heavier load, we have an increased latent period. But then we have a decreased duration of contraction and a decreased velocity of shortening. Okay, and all that means is the heavier the load, the longer it takes to lift it, the shorter the duration we can actually lift it, and the longer it takes to warm the machinery up to actually overcome this load. Okay, all right, so that's what we spoke about in terms of factors that affect the force generation. Let's move on to talk about the ATP source of muscle contraction. Okay, so we spoke about 3 sources of ATP for skeletal muscle production. What were those 3 sources? Oxidative phosphorylation, what else? Oxidative phosphorylation, what else? What was that transient source that kind of got things started, got things warmed up? The creatine phosphate system, right? Creatine phosphate. And then what was the other source? Anaerobic glycolysis. Perfect. Anaerobic glycolysis. This is sometimes called substrate level phosphorylation. I'll make a note here. Sometimes called substrate level phosphorylation. Okay, now let's describe these a little bit. What are the principles of oxidative phosphorylation? What are the principles or the features of oxidative phosphorylation? First of all, we know it requires oxygen. What else? How much ATP does it give us? Does it give us an efficient source or is it not an efficient source? It's an efficient source, right? So we got a lot of ATP. How long does it take to warm up? Does it take more time or less time? More time. It takes more time, right? It takes significantly more time than, say, anaerobic glycolysis. Okay, but it requires oxygen. As far as anaerobic glycolysis, we know that it gives us less ATP. It takes less time. And what else does it give us? It doesn't require oxygen, but what else does it give us? What's the catch with anaerobic glycolysis? Lactic acid. Perfect. So we got lactic acid buildup. Okay, so it takes less time, gives us less ATP, but it's going to give us lactic acid. Oxidative phosphorylation is going to take more time, it's going to give us more ATP, but we're going to need to have oxygen, meaning that we're going to need to have a continuous ventilation. And then creatine phosphate, we know the enzyme responsible for this sort of transient pathway was what? What's the enzyme that helps give us that transient source of ATP from creatine phosphate? Creatine kinase, right? So that enzyme creatine kinase gives us that transient ATP. When I say transient, I mean it's only a temporary source while the other pathways get started. Now, as far as understanding the 2 types of exercise, we talked about light intensity exercises versus heavy intensity exercise, and what was the order of recruitment from these sources for light exercise? So for light intensity exercise, we could maintain ventilation. So what's the first thing that we tap into? Well, before anaerobic glycolysis, what do we tap into? For all muscle contraction, what's the first source of ATP? Creatine phosphate. Perfect. So we start with creatine phosphate, and then where do we go from there? I think I'm hearing in the back. Then we go to oxidative phosphorylation, right? And that's going to be the main source of light intensity exercise, okay? The main source of light intensity exercises. Why? Because we are exercising at a rate where we can maintain oxygenation, we can maintain ventilation, and so this is the main source of light intensity exercise. What about heavy intensity exercise? We, again, start with creatine phosphate, but then where do we go from there? Can we maintain oxygenation? No, we can't maintain ventilation, so we don't tap into oxidative phosphorylation as much. What's the main source here? Anaerobic glycolysis, right? Anaerobic glycolysis. And then that's going to give us lactic acid. Remember that as a byproduct. Okay. All right. So that's pretty much all we talked about for muscle in the way of force generation and in the way of ATP. Now I won't go into the details of the types of fibers. We went over that extensively. And we even went over this table. Make sure you review this table, right? The different types of fibers. Make sure you know why we have small diameter versus large diameter, why we have mitochondria versus no mitochondria, why we have capillaries versus no capillaries, okay? What are the various oxidative capacities for all three of these, all right? Make sure you also understand the fiber types based upon, or the recruitment, the size principle here, right? How do we recruit fibers based upon their states, their features? So we recruit oxidative fibers first. Why? Based upon the size principle, why do we recruit oxidative fibers first? They have a smaller diameter. They have a smaller fiber diameter. They're likely attached to smaller motor units with smaller axon diameters, et cetera, et cetera. So we recruit these types of fibers, then these types of fibers, and these types of fibers. Which of these fibers do we use for long endurance, slow intensity exercises, muscles like the soleus muscle? Which of these fibers? Slow oxidative, right? Which of these fibers do we see in maybe the biceps or the hamstrings or the gastrocnemius, right? For power and strength. The fast glycolytic fibers, right? Make sure you understand that and understand why, okay? All right, I'm not gonna go into any more detail on that bit. The next thing we did was smooth muscle, and we talked a lot about the excitation contraction coupling of smooth muscle. You should already have this drawing. And this is a really important place to sort of study the general facets of muscle contraction. Make sure you understand why calcium is regulating, how calcium is regulating smooth muscle contraction versus how calcium regulates skeletal muscle contraction, right? And make sure you understand calmodulin and its role, and then MLCK and its role here as well. Okay, and so I won't go into the steps in detail because we talked about this, but understand how this compares to skeletal muscle contraction in terms of the fact that there's no sarcomere here, right? In terms of the fact that the source of calcium is extracellular as well as the SR, okay? And I hope you all have drawn that table that compares those three types of muscle: skeletal muscle, smooth muscle, and cardiac muscle, and look at the differences, et cetera, et cetera. Okay, any questions on this? Okay, and I think that's all we looked at for, is it? Yeah, for muscle. Mm-hmm.
So calcium still is the driving factor, whether it's flying it on, will determine cross-bridge cycling, and then if it's off, it stops cross-bridge cycling.
Yeah, yeah. So it's going to bind to calmodulin, and that will turn things on or turn things off. Okay. Where does it bind in skeletal muscle and cardiac muscle? To actin, what part of actin? Troponin, right? Binds to troponin and does that whole shifty thing with tropomyosin, and it regulates that way in cardiac and skeletal muscle. In smooth muscle, it also regulates, but in a different way. It binds to something different, brings about a different enzyme, which then brings about cross-bridge cycling or not, okay? All right. So that was it for muscle. So that's all we did for muscle. That was muscle packet 1, muscle packet 2. And there was a really nice study table at the end that compared single unit versus multi-unit. That's also something that we talked about. And make sure you look at that. Make sure you understand the difference between single unit, multi-unit, and skeletal and cardiac, okay? There are 2 types of smooth, and there's skeletal and cardiac as well. Any questions on muscle? Okay, now I got a few questions asking specifically about the difference between series and parallel flow, and then asking about some of the valves, right? So I'll discuss those, and then I'll sort of go into some more general things. So when we talk about series and parallel flow, let me see. Okay. We talked about this idea of the heart, the body, which is all of our peripheral tissues, back to the heart and then to the lungs. This is the way blood flows in our body, and this is a must, okay? Fresh oxygenated blood must leave the heart, travel through arteries and arterioles. Make sure you know this pathway, right? Arteries to arterioles to capillaries. Then from capillaries, we get to venules, to veins, then back to the heart, okay? And then from the heart, it needs to go to the lungs via pulmonary arteries to get reoxygenated, and then back to the heart via pulmonary veins to get pumped back through the circulation, okay? So the question was, how do we differ between series flow and parallel flow? So series flow includes the circulation that must happen. Blood must go from the heart to the body, back to the heart, to the lungs, to the heart, etc. Parallel flow happens when we get to the peripheral tissues of the body in terms of our capillary beds, and blood can go to the liver, some of it can go to the kidneys, some of it can go to the bladder, and so we call this style of flow parallel flow because all of the blood does not go to each of these areas. The blood splits off here, and we have, say, 20% going to the kidneys, 20% going to the liver, 20% going to the bladder, et cetera, et cetera. And so this style of flow where the blood has an option is called parallel flow versus this flow where the blood does not have an option. This is called series flow. Okay, we also have a bit of parallel flow in the lungs. When blood gets into the pulmonary arteries, half of it can go right and half of it can go left, right? We have 2 lungs. And so this will also constitute parallel flow. So whenever that blood has an option, right, when it's dividing, some of it can go here, some of it can go there, that is parallel flow, whereas the general circulation of going from heart to body, back to heart to lungs is series flow. This blood does not have an option, right? It has to go through each of these. I hope that made sense. Any questions on that? Okay, so that was series versus parallel flow. The next question that I got had to do with the valves and understanding which valves are where and what they're doing. And so we'll kind of look at this drawing here. So if I can zoom in. Oops. Okay. Just trying to zoom in, but I can't. Okay. So just to describe, we have 2 semilunar valves and 2 AV valves, right? The semilunar valves are the valves that are exiting the heart, right, from the ventricle out into the vessel. The AV valves are the valves that are between the atria and the ventricle, okay? On the left side of the heart, we have the bicuspid valve. On the right side of the heart, we have the tricuspid valve. Now, in terms of the general circulation, blood enters the heart through the vena cava, right? This is all of our deoxygenated blood that's coming back. It's going to go through the right side of the heart, the right atrium, the right ventricle, then out through the pulmonary arteries. Here it's going to go to the lungs. It's going to come back from the lungs into the pulmonary vein, back into the left side of the heart, then go to the left ventricle, then out of the aorta, out to our peripheral tissues. Okay, now that happens over and over again. So as far as the location of the valves, we have our semilunar valves that are leaving the ventricles, right, from ventricles to the vessel, whereas our AV valves are going from the atria to the ventricle, okay? And I hope that answered that question. So the location of the valves and then the general flow through the heart. Now, just to remind ourselves here what's happening in terms of what's open and what's closed. During systole, we have ventricular contraction or ventricular emptying or ventricular ejection. Now, what phases of the cardiac cycle correspond with systole? There are two phases that correspond with systole. So we got our four phases: ventricular filling, isovolumetric contraction, ventricular ejection, and isovolumetric ventricular relaxation. So we said what two phases correspond with systole or emptying? Phase 2 and 3, right? So the isovolumetric ventricular contraction, which is phase 2, and then the ventricular ejection, which is phase 3. And so basically what's happening there is that in phase 2, the chamber is contracting. Remember, all valves are closed at this point. This is isovolumetric. The chambers are contracting. That's phase 2. And then in phase 3, the pressure now is high enough in the ventricle that it can open up these semilunar valves, and then we have ventricular ejection. So those two phases, phase 2 and 3, correspond with systole or ventricular emptying, okay? On the other hand, there's diastole, and diastole is ventricular filling. This is where the chambers are accepting blood, and that corresponds with phase 4 and phase 1. Phase 4 is isovolumetric ventricular relaxation. This is where, again, all four valves are closed. The ventricle is now relaxing. The pressure is now falling. And then phase 1, which is ventricular filling, which is where the valves now open. And the valves open because the pressure now is dropped below the pressure in the atria. And so we have this valve opening, and we can resume the rest of ventricular filling. And so those two phases, phase 4 and phase 1, correspond with diastole, which is ventricular relaxation or ventricular filling. Now remember, the heart spends more time in diastole, it spends more time filling. And that's because it needs to fill adequately in order to eject. In order for us to get that adequate stroke volume, the heart must spend enough time filling or in diastole, okay? Let's look at what valves are open and closed down here. So we know that the AV valves are open when the atrial pressure exceeds the ventricular pressure. We talked about this a little bit, right? The pressure is what really creates the valve opening, that pressure gradient. So the AV valves will only open when the pressure here is lower than the pressure here, okay? Likewise, the semilunar valves will only open when the pressure here exceeds the pressure in the vessel, the pressure out here. So the valves here, right, are pulmonary on the right side and aortic on the left side. These valves only open when there's high enough ventricular pressure to actually force these open. This is a passive process, okay? All right, any questions on that? Any questions on that bit? Make sure you understand the phases of the cardiac cycle, okay? I'm not gonna go over them in detail, and what valves are open during each phase. So yeah, both vena cava, we have the superior and inferior, and they're coming into the right atrium. Any other questions? Any other questions? Okay. I hope that means that everyone has this down packed and is ready for Friday. Okay. The next thing I will go over, I didn't get questions on this, but I'll just touch on it briefly, is the conduction system here. Okay, and if you have questions on it, you can feel free to point those out. But we talked about the conduction system. Okay, make sure you know that the impulse is traveling from the SA node. It's actually going through the intranodal fibers here. Right, it's gonna go through the intranodal fibers, which are part of our conduction fibers. Then it's gonna go to the AV node, then the bundle of His, then our bundle branches right and left, and then the Purkinje fibers, and it's gonna squeeze that impulse up, okay? Also make sure you know that the purpose of the AV nodal delay is to ensure that the atria contract fully before the ventricles contract. That's really important. Both atria must contract fully, and then both ventricles contract. Okay, so the sequence of the impulse as well as the sequence of the actual contraction of the chambers. Any questions here? Any questions on this bit? Okay. And I don't think I have any other specific questions or any other specific areas. Are there any specific areas that you guys would like me to touch on, to review? Other than that, I have a few practice questions, which are mostly on the cardiovascular system. But if you have other areas that are still confusing that you'd like me to just touch on or review, I can do so now.
The 5 phases of electrical activity in cardiac cells,
Oh, okay. Yeah. So in the contractile cell, well, actually, let's look at both. So remember we said that we have 2 types of cells in our heart, right? We have contractile cells, which are actually doing the work, the contraction. And we have pacemaker cells that are basically just setting the rhythm. They're not so much contracting as they are setting the rate and the rhythm of the heart, okay? And so we looked at the phases of contraction in a contractile cell. And this is basically, before we get into the detail, this is basically what's happening in terms of the channels on a contractile cell to bring about an action potential in this cell, okay? And so when we start out, we start at rest. And this is how a contractile cell is at rest. We said it's a little bit more negative at negative 90. And we have the potassium channels more open than sodium and calcium. And all that this means is that we have a more or an increased permeability for potassium, more doors, than we do for sodium and for calcium, okay? And so from that, the first thing that happens is the opening of sodium channels. And so opening of sodium channels depolarizes the cell, sodium rushes in, and that's responsible for generating the action potential, this upstroke. That's phase 0. After that, we still need to maintain depolarization for a little bit. So when we close sodium channels, so phase 1 is closing those sodium channels, this is an attempt of the cell to begin repolarizing. But we do not repolarize right away. We need to sort of delay this process. What did we say was the importance of delaying repolarization in a contractile cell? What's the importance of delaying or interrupting this repolarization? What's happening during this delay? Calcium, right? Calcium needs to get into the cell. And so we interrupt repolarization by opening up our calcium channel and closing our potassium channel. So in this plateau phase, we briefly interrupt repolarization in order to facilitate the movement of calcium into the cell. Calcium is responsible for binding to troponin, right? Regulating crossbridge cycling and bringing about contraction in this contractile cell. Mm-hmm.
So in phase 1, there's some potassium in there.
Potassium is still coming in. We haven't interrupted potassium flow. We start with potassium channels open. We haven't interrupted that, right? Good question. Any other questions? Oh, I'm sorry. Is that better? Okay. Any other questions? So that was a good question. So we start off with potassium channels normal. We haven't interrupted potassium flow until phase 2. And this is to stop or halt the repolarization process. And the reason why is to facilitate calcium movement to help get that crossbridge cycling going. When we're done here, we've delayed this enough, right? We then flip-flop the orientation of those 2 channels. So instead of having calcium channels open and potassium channels closed, in phase 3, we now have potassium channels open and calcium channels closed, and that continues the repolarization, which brings our membrane potential right back down to rest, okay? Because in this phase, we don't have calcium moving anymore. We've closed those calcium channels. And so what we have now is only the movement of potassium. And we know from prior knowledge that potassium is rushing out, right? That's ECDF. And so it's going to make the inside of the cell more negative. And so the membrane potential reestablishes back to rest, which is negative 90 or so. Okay. Any questions on that? I hope that answered Any confusion? Was that your question, Tom? Okay, no, it's okay. Yeah, so this was in the contractile cell. Let's take a look at the pacemaker cell really quickly. So in the pacemaker cell, we had a different sequence of events, okay? We had a gradual depolarizing here, a gradual depolarization happening here. We start off at -70. We open up our funny channels, and I talked about the funny channels being mainly sodium channels. So sodium is the main ion that's moving through, in terms of the net movement of ions that's moving through funny channels. So we open up funny channels, and then we close potassium channels. So from rest, right away, we close potassium channels, and that helps depolarize this cell. Now, the excess sodium that starts to accumulate further triggers the opening up of fast calcium channels. And fast calcium channels open, and that's what gets us to threshold. Those T-type, those transient or fast calcium channels gets us to -55. And from there, we further open up calcium channels, a different type of calcium channel here. It's a slow calcium channel, and that is responsible for the upstroke, for the rapid depolarization of a pacemaker cell. Okay, now when we rapidly depolarize the cell, then we close those calcium channels and we open up potassium channels. And remember, we started with them closed, so now we can reopen them, and that facilitates repolarization. Okay, so important to distinguish that in a pacemaker cell, the ion that's responsible for depolarization is calcium, specifically through a slow calcium channel, whereas in a contractile cell, the ion that's responsible is sodium, and that movement of sodium is the first thing that happens in the cell, whereas in a pacemaker cell, there's a gradual depolarization.